In this video, we will discuss about different translatome profiling method. So, in our previous videos, we have discussed one of the method that is ribomap that is used for translatome. So, in this video, we will cover a brief overview of how this translatome method works before those precise ribomap star map techniques arrived. So, there are three basic approaches that are shown here. First one is that they will use the polysome method. In this, what they will do is they will use the cell extract and pass them through different sucrose gradient to in order to separate the free RNA or 40s or free RNA, that is 40s RNA, 60s RNA, 80s RNA, that is not bound to any free RNA, and this 40s, 80s, 60s is the part of ribosome, and these free RNAs and ribosomes are being separated out through this fractionation using the sucrose gradient from 10 to 15%. And after that, this polysome, that is the RNA that is bound to the ribosome, is then further separated out. Later on, the microarray assay of this polysome will help us to know which are the RNAs that are bound to the ribosome. As this is based on the assumption that initiation of the mRNA translation is the most critical step for the translation process. So if most of the RNAs that are bind to the ribosome are considered here as the mRNA that are already been initiated for translation. So this can be determined from the microarray assay of the polysome, and simultaneously they can also do the RNA-seq of the polysome's RNA. From this they can find out the abundance of the mRNA that are present in the polysome, and they can be later be inferred, or they can later be cross-verified from the literature whether those RNAs are overexpressed in these samples or not. So, and this is one of the approach. And next approach is that to use this RNase one digest. In this, we are separating based on sucrose gradient. In this, what we will do is we will digest the RNAs that are not protected, or that are not protected means that are not covered by the ribosome, like this. Except this part, all other RNase part are been digested by this RNase I, and after that they will do the sucrose selection through this electrophoresis, and from this they will isolate this RPF part, or we can say uh, ribosome protected fragment, which is of around 30 nucleotides, and then this RPA is then ligated with the primers, so that this reverse transcriptase can be carried out on this RPF, and cDNA can be formed, and all this reverse transcriptate or RNA depletion and this amplification process can be carried out, and later on the RNA-seq data of this RPF will then help us to know which are the RNAs that are being bind to the ribosome. So those RNAs can be considered as those RNA can be considered as more in those RNA, or we can say those RNA can be considered as the are in the translated, or in the translation step, or are more translated compared to the RNAs that are not detected in the RNA-seq data. Last one is to use the affinity tag in order to purify affinity tags such as GFP or protein A tag in order to purify the proteins that are bind to the in order to purify the mRNA that are binds to the ribosome. So the difference in all these three steps is the purification step. In this, the sucrose gradient is used. In this, The RNase one is used to digest the unprotected RNase, and in this they have used affinity tags such as GFP or protein A tag to separate out the mRNA that is bind to the ribosome. Later on, the wash and elution step will carried out in order to filter out the RNAs, in order to filter out the RNAs that are bind to the ribosome from the other cell extracts. And later on, the this microarray can be performed as it is performed in the polysome profiling to find out the ribosomes, to find out the mRNAs that are bind to the ribosomes. And in all this step, this RNA-seq can also be introduced in order to improve the efficiency. For example, in polysome, along with the microarray, this RNA-seq data can also be carried out to further substantiate their method. And similarly, in this affinity purification method also, along with the microarray, the RNA-seq data can be carried out simultaneously in order to substantiate the finding from the ribosome affinity purification. This is the basic overview of how the translatome profiling method emerges in the field, and which then later on been converted and later on been formed into a more scientific, or we can say accurate methods such as ribomap and star map. And among this ribomap we have covered in our previous videos. If you need to understand more about this translatome profiling methods in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.